Hello, and welcome. Today, we are going to explore a fascinating intersection of material science, optics, and artificial intelligence. We'll be taking a deep dive into a review paper by Rigoberto Adventula and Jihua Chen, titled AI-ML-Driven Surface Plasmon Resonance and Spectroscopy. This work charts a course from the fundamental principles of a powerful sensing technique to the cutting-edge vision of fully autonomous, AI-driven laboratories. The central theme is the evolution of a technique called Surface Plasmon Resonance, or SPR. We'll see how it has moved from foundational studies of how molecules stick to surfaces, known as adsorption-desorption kinetics, to incredibly sophisticated sensors. These sensors can be built using what are called Electropolymerized molecularly imprinted polymers, or E-MIPs for short, which are essentially custom-designed molecular traps. The authors highlight their own extensive research in this area, focusing on the optical properties and electrochromism, that is, the color-changing ability, of specialized polymer materials, including ultrathin films and unique structures called polymer brushes. But the real transformative leap discussed here is the impact of artificial intelligence and machine learning, or AI-ML. We'll explore how these computational tools are revolutionizing data interpretation and leading to the conceptualization of what are known as self-driving labs. These are not just automated systems, they are autonomous systems that can design, execute, and learn from experiments in a closed loop, accelerating the pace of discovery. A key point is that for these AI systems to learn effectively, they need vast amounts of high-quality training data, and the authors propose that high-throughput experimentation using SPR can provide exactly that. Ultimately, this work presents a vision for the future of material science, where conventional experimental design is integrated with AI-driven sensing to rapidly discover and optimize the next generation of sensors for everything from materials research to biomedical applications. So let's begin by understanding the core technology here, surface plasmon resonance.
A surface plasmon, or a surface plasmon polariton, is essentially a wave of electron density that forms at the interface between a metal, like gold, and a dielectric material, which could be air, water, or a specific chemical layer. These waves are special because they are spatially localized, confined to that tiny interface, and they can produce highly enhanced electromagnetic fields. This behavior is described by a fundamental dispersion relation, an equation that connects the wave vector of the plasmon to the optical properties of the materials involved, specifically their relative permittivity and permeability, as well as the frequency of the light. The key to SPR's power as a sensor is that the conditions needed to create this resonance are incredibly sensitive to the environment right at that interface. Any change, like molecules binding to the surface, alters the local dielectric properties. This in turn shifts the resonance conditions. By measuring that shift, we can detect and quantify biochemical events in real time. A common way to harness this phenomenon is with an experimental setup known as the Kretschmann configuration. In this setup, light is shown through a prism onto a thin metal film, typically gold, deposited on a glass slide. At a specific angle of incidence, under conditions of total internal reflection, the light energy couples with the electrons in the metal film and excites a surface plasmon. This coupling causes a sharp drop in the intensity of the reflected light. The angle at which this minimum occurs is the SPR angle.
A typical experiment, as illustrated in the paper, might involve a sensor chip coated with gold, onto which antibodies are attached. When a sample containing the target biomolecule is introduced, these molecules bind to the antibodies. This binding event adds mass to the surface, changes the local refractive index, and causes the SPR angle to shift. A detector monitors the reflected light and tracks this change in the resonant angle over time. The resulting plot, often called a sensorgram, shows the association of the molecules binding, a steady-state equilibrium, and then the dissociation as the molecules unbind. This allows for non-invasive, real-time monitoring of molecular interactions. The physics behind this, the layered dielectric models based on Maxwell's equations, are very well established. This solid theoretical foundation means we can reliably model the system using methods like the transfer matrix method. And because the fundamentals are so well understood, SPR can be combined or hyphenated with other techniques like Raman spectroscopy, infrared spectroscopy, or even microscopy to get even more information. This is where machine learning enters the picture. Given the well-established physics, ML algorithms can be trained to improve the throughput, performance, and applicability of SPR. Instead of relying solely on explicit programming and traditional models, ML uses data-driven methods. It can recognize patterns and trends in massive datasets that might be invisible to a human observer. It can be used to analyze data, extract meaning, and even design new plasmatic structures with desired optical properties. This concept of inverse design, where you start with the desired outcome and have the AI design the material or structure to achieve it, is incredibly powerful. Deep learning, a subset of ML, can push this even further, exploring novel plasmonic structures and instrument configurations that go beyond conventional designs. The scientific literature is filled with examples of SPR being used for sensing, particularly in biosensing. The applications are widespread, from pharmaceutical research and drug discovery to clinical diagnostics like point-of-care testing and environmental monitoring for pathogens or pollutants. It's also a powerful tool for material characterization, allowing scientists to measure the optical constants of thin films, monitor the layer-by-layer assembly of materials, and study corrosion or polymer swelling.
Let's look at some of the specific analytical advances the authors review. One of the earliest uses of SPR was for the in situ, meaning in place and in real time, investigation of how polymers in a solution adsorb onto a surface and self-assemble. This allows researchers to study the kinetics of these processes. For example, SPR has been used to watch the layer-by-layer self-assembly of complex organic molecules that are relevant for high-performance organic transistors. Another key area is the development of non-fouling surfaces, which are critical for biomedical implants and sensors that need to resist having proteins and other biomolecules stick to them indiscriminately. The authors describe studies where SPR was used to test coatings made from OEG-agylated dendron carbazols. By monitoring protein adsorption kinetics, they could determine that the second generation, or G2, dendrons showed the best resistance to protein fouling. Building on this, the development of highly specific sensors is a major goal. This is where those electropolymerized molecularly imprinted polymers, or E-MIPs, come in. The process of molecular imprinting is conceptually like making a keyhole for a specific molecule. First, a functional monomer, the building block of the polymer, is allowed to form a complex with the target analyte, the molecule you want to detect. Then, this complex is electropolymerized, meaning an electrical potential is used to grow a polymer film directly onto the sensor electrode, trapping the analyte within the polymer matrix. Finally, the analyte, or template molecule, is washed away, leaving behind a cavity that is perfectly shaped and chemically complementary to it. When a sample is introduced later, only the target analyte can fit snugly back into this molecular memory site, generating a highly selective sensor signal. The authors detail their extensive work in this area. For example, they developed E-MIPs for SPR-based sensing using electroactive monomers like terrathiophene and carbazole. In one study, they created a sensor for theophylline, a drug used to treat respiratory diseases. The process involved creating a polyterrathiophene film with imprints for theophylline. When theophylline was reintroduced, it would rebind to the imprinted sites, causing a measurable shift in the SPR signal, allowing for highly selective and sensitive detection compared to other similar drug analytes. They combined E-MIP sensing with another technique called electrochemical impedance spectroscopy, or EIS, to create an extraordinarily selective sensor for bisphenol A, a common industrial chemical. They also developed sensors for dopamine, a crucial neurotransmitter, using a different type of E-MIP made from poly-P-aminostyrene. Their work has even extended to detecting much larger analytes, like the protein epitope for the dengue virus, demonstrating the versatility of the E-MIP approach. Beyond just creating selective binding sites, the authors have also deeply investigated the fundamental optical and electrochemical behaviors of the polymer films themselves. They developed a powerful experimental setup that combines electrochemistry with SPR, often referred to as EC-SPR. This allows them to simultaneously apply an electrical potential to a polymer film and monitor the resulting changes in its optical properties in real time. For instance, they conducted in situ studies on the electrochemical polymerization of polyaniline, or PANI. They could watch the film grow and study its redox behavior, how it responds to being oxidized and reduced, all while measuring changes in its dielectric properties via SPR. They applied the same combined approach to study other important conducting polymers like PEDOT, which is widely used as a hole transporting material in organic electronics, like OLEDs. By using two different laser wavelengths in their SPR setup, they were able to distinguish between changes due to the polymer's electrochromic behavior and those from the movement of dopant ions from the electrolyte. They've even pushed the hyphenation further, creating a 3-instruments-in-1 setup that combines electrochemistry, SPR, and atomic force microscopy, or AFM. This allowed them to simultaneously observe the electrochemical processes, the optical response, and the changes in surface morphology and topography of a conducting polymer film as it was being grown.
This detailed characterization extends to how these films are constructed. The authors reviewed two key self-assembly methods for creating ultra-thin polymer films. The first is the layer-by-layer, or LBL method, where a charged substrate is alternately dipped into solutions of positively and negatively charged polyelectrolytes, building up a multi-layered film one layer at a time. The second is the Langmuir-Blodgett, or LB technique, where amphiphilic molecules, molecules with both a water-loving and a water-hating end, are spread on a water surface, compressed into a monolayer, and then transferred onto a solid substrate. Their work has shown how EC-SPR can reveal changes in nanostructure and dielectric properties in these highly ordered films, which is critical for applications in solid-state devices. A particularly interesting application they explored was using gold nanoparticles to enhance and tune the SPR signal. They investigated a system with an AU nanoparticle and polyelectrolyte layer-by-layer film. They found that the system showed pH sensitivity, with the plasmonic hotspots between the nanoparticles being dependent on the distance between them, which could be controlled by the swelling of the polymer. Their computational models, based on Maxwell-Garnett theory, matched well with the experimental results, showing how the filling factor of nanoparticles in the film could be used to tune the SPR response at different laser wavelengths. Another major area of their research involves polymer brushes. Unlike a simple flat film, polymer brushes are made of polymer chains that are tethered at one end to a surface, often at high density, causing them to stretch away from the surface like the bristles of a brush. These structures are distinct from self-assembled monolayers, or SAMs, which are typically just a single molecule thick. Polymer brushes can be made from a wide variety of polymers. Their thickness can be tuned, and they can have more complex architectures. The authors have used SPR, both in situ and ex situ, to characterize the growth of these brushes from surfaces, for example from clay nanoparticles or directly from gold substrates, using various surface-initiated polymerization techniques. This control over polymer brush length and grafting density is essential for their potential use in biomedical devices and sensing. A diagram in the paper shows how these brushes can be fabricated either by a grafting-to approach, where pre-made polymers are attached to the surface, or a grafting-from approach, where the polymer is grown directly from initiator sites on the surface. These structured polymer brushes can even be used to regulate the differentiation of stem cells. So we have this incredibly versatile and sensitive analytical platform in SPR, capable of studying everything from fundamental molecular interactions to the behavior of complex material architectures. This brings us to the next, transformative part of the story, the integration of artificial intelligence and machine learning. Machine learning is applied to both simulation and experimentation.
The paper provides a helpful taxonomy of the main ML algorithms. Broadly, they are divided into supervised learning, where the algorithm learns from label data to make predictions, and unsupervised learning, where the algorithm explores unlabeled data to find hidden patterns or structures. Supervised learning methods include familiar statistical tools like linear regression, but also more complex ones like support vector machines, SVMs, and neural networks. Unsupervised learning includes methods for clustering data, like k-means, or for reducing the dimensionality of complex data, like principal component analysis, PCA. The chart presented shows a spectrum of these methods, ranging from simpler, more interpretable models like linear regression and decision trees, to more advanced, high-performance algorithms like deep neural networks, which can be less interpretable, a sort of black box. Deep learning is a subset of ML that uses artificial neural networks, or ANNs, with many layers. These are powerful for tasks like classification and regression. There are different types of ANNs. For example, convolutional neural networks, or CNNs, are particularly good for image analysis,
and generative models like generative adversarial networks, GANs, and variational autoencoders, VAEs, can actually generate new synthetic data that resembles the real data they were trained on.
The promise of applying these ML workflows to plasmonics is to accelerate scientific discovery. The goal is to use supervised, unsupervised, and deep learning across every aspect of the SPR experiment. For instance, ML can analyze vast data sets of sensor responses to identify the optimal settings for sensitivity and selectivity. Traditional physics-based modeling, while powerful, can be slow and computationally expensive, especially with many variables. ML models can act as fast surrogate solvers for the complex electromagnetic equations, drastically reducing simulation time and enabling rapid optimization of things like signal-to-noise ratio. There is a large body of existing SPR data that can be used to train and fine-tune these models. Neural networks and genetic algorithms can enhance the design of complex plasmonic nanostructures, optimizing their shape, size, and arrangement to achieve very specific optical responses, like a particular color or emission wavelength. By using deep learning for parameter tuning, scientists can create digital twins of their sensors to predict optimal geometries and responses before ever stepping into the lab.
This leads us to the concept of self-driving labs. The authors present several recent examples of these ML-driven workflows in action.
In one case, a deep learning method using a CNN was developed to improve the precision of SPR angular scanning. Instead of complex post-processing or polynomial curve fitting, the CNN was trained on simulated reflectance spectra to directly and accurately locate the plasmonic angle. The flowchart for this process shows how a large simulated dataset is created using Fresnel equations, with various types of noise added to make the model robust, which is then used to train the network. In another example, an ML-driven sensor using carbon nanotubes and graphene was modeled and built to detect formalin and water. Regression-based ML models have been used to pinpoint SPR sensors for enhanced detection of SARS-CoV-2 particles. Deep neural networks have been used to establish the complex relationship between a nanoparticle's dimensions and its far-field and near-field optical spectra. And in a material synthesis context, a genetic algorithm-driven artificial neural network was used to find the relationship between synthesis variables and the resulting surface plasmon wavelength of gold sea urchin-like nanoparticles, which have promise for cancer therapy. Perhaps the most powerful application is inverse engineering.
The paper shows a schematic of ML being used for data analysis, where it can perform tasks like dimension reduction and clustering, or classification and regression to help with detection and diagnosis. Going a step further, it showcases an architecture for a GAN-based network for designing a gold film structure. Here's how it works. A generator network proposes a structure. A simulator network, which is a pre-trained physics proxy, predicts the optical spectrum of that proposed structure. This predicted spectrum is then compared to the desired target spectrum. A third discriminator network then tries to distinguish between real data and the generated data. Through this adversarial process, the generator gets progressively better at designing a structure that produces the exact optical signal required. This is a complete closed-loop design process driven by AI. Another striking example is the AI-aided detection of SARS-CoV-2 using localized surface plasmon resonance. The work details the fabrication of a sensor chip with an array of gold titanium dioxide nanocup structures. When the virus is present, the optical properties of the sensor change. An AI classifier model is trained on microscope images of the sensor with and without the virus. The system uses PCA to visualize variations in the data from different virus concentrations. Then, models like logistic regression and a support vector machine are trained to classify a new image as either positive or negative for the virus, with extremely high accuracy, greater than 97%. The confusion matrix shown for the SVM classifier demonstrates this power, correctly identifying hundreds of positive and negative cases with very few errors. So, what does the future hold? The authors conclude by outlining 4 key areas for future work. First, predictability with ML methods. The future is in hyphenated instruments that combine SPR with fluorescence, electrochemistry, and microscopy. ML will be essential to analyze the complex, high-dimensional, multimodal data from these systems, increasing their reliability and accuracy. Second, ML methods for new dielectric materials. On the material side, ML-optimized molecular dynamic simulations can be used to understand the complex interactions at the sensor interface. This can help design new organic dielectric materials and even build multi-parameter sensor models that account for environmental factors like temperature and humidity. Genetic algorithms and ANNs will guide the actual synthesis of these new materials. Third, real-time data and fast computational resources. A major challenge is that ML is only as good as the data it's trained on. Generating the massive, well-parameterized datasets required is critical. This necessitates high-throughput experimentation, ideally from an autonomous self-driving lab setup, coupled with high-performance computing to enable real-time simulation and analysis. And fourth, nanoparticles, metamaterials, and photonics. The possibilities for plasmonic structures go far beyond the simple thin films used in basic SPR. Using ANNs and inverse design principles, researchers can design complex nanostructures, nanoparticles, and even metamaterials with exotic optical properties. This is a major frontier where materials discovery and AI will grow together. In conclusion, the work of Advincula and Chen maps out a compelling trajectory. It starts with the well-established and powerful sensing technique of SPR and shows how it can be enhanced and ultimately transformed by the integration of AI and machine learning. The vision is one of a virtuous feedback loop. Automated experiments generate vast amounts of high-quality data, which trains more powerful ML models, which in turn design better materials and more efficient experiments. This leads to the ultimate goal of the self-driving laboratory, a paradigm that promises to dramatically accelerate the pace of discovery in material science, chemistry, and biomedicine.
The paper itself was last updated on February 16, 2026, indicating it is a forward-looking perspective on this rapidly evolving field. Thank you.